Good afternoon, everyone. Welcome to the Wednesday afternoon lecture series. To those of you here in Missouri and to many others who are listening in the videocast. These are our special opportunities in the middle of the week to hear from scientists who's doing exciting work that I think often enlivens our experience and sometimes suggests new ideas to all of us that have a chance to soak up what's being presented. We're fortunate today to have as our speaker Dr. Amy Powler, who comes to us from Northwestern, where she is now the professor and Walter J. Hamlin Chair of the Department of Dermatology, as well as being professor in the Department of Pediatrics, residency director for Department of Dermatology, and an attending at the Ann and Robert H. Lurie Children's Hospital of Chicago. Amy got her undergraduate degree at Brown, did a master's in genetics, also at Brown, and then went on to get her MD at Stanford. And following residencies in pediatrics and dermatology at Northwestern, and a postdoctoral fellowship in a place near and dear to my heart, UNC, where she worked with Robert Brigham. She then went back to Chicago, where she has been in a variety of increasingly significant leadership roles to her current position there at Northwestern University's Feinberg School of Medicine. Her research has consistently focused on areas of dermatology that are exciting and cutting edge, both uncovering genetic causes for certain skin diseases, but also very invested in coming up with therapeutic strategies, including pretty exciting exotic things like nanoparticles that are loaded up with siRNA. Maybe she'll even say something about that. She's also been a dedicated servant to the NIH, serving on study sections. Thank you, Amy, and currently serves as a member of the NIAMS Advisory Council. I'm sure Steve Katz is grateful for that. And she's a long-term grant recipient. We would love to be able to say that her grant support will be as stable as it possibly can be. Of course, our current environment makes that a little tougher, but she's obviously been very successful, and I suspect will continue to be. So it's a great pleasure to be able to have her here this afternoon. Please join me in welcoming Dr. Amy Powler.
Thank you very much. I'm so honored to be here today and to talk with you a little bit about genetic skin disorders and where we're going in new therapies that can be applied to this patient population. I'm going to start by just telling you that when I talk about genodermatoses, I am talking about the range of genetic skin disorders, and it turns out that about 20% of monogenic disorders involve the skin, hair, and or nails. Now, these are highly visible, usually life-altering and sometimes life-threatening disorders, and I'm going to talk largely about two groups among the many groups of disorders that are genetic that have these manifestations. I'll be talking about blistering disorders and the inherited group called epidermolysis bullosa, or EB, and I'll be talking about scaling disorders, the group that's been called the ichthyoses. So our progress in understanding genetic disorders of the skin really paralleled that of understanding other genetic diseases, and we know that really the explosion of information started in the late 1980s and early 1990s, with gene discovery largely based on linkage analyses, animal models that were generated, and then confirming candidate genes by restriction digest and sequencing. And among the genodermatoses, really the first structural one that was discovered was one in which I had the pleasure of being involved in that genetic discovery, one called epidermolysis bullosa simplex, or EB simplex, an autosomal dominant blistering disorder. And this was really through the work of Elaine Fuchs's lab. Now, we knew that this was a disorder that involved the basal keratinocytes because of the fact that ultrastructural studies from the late 1960s had shown that, in fact, there was cleavage through the basal keratinocytes with an intact basement membrane zone, intact hemidesmosomes, and a cleavage that was above that through the keratinocyte with abnormal-looking keratin tonofilaments. We could also see that by immunofluorescence, that there was abnormalities in the appearance of these keratin tonofilaments. And the lab then was able to do what we used to do to discover gene defects, and that is gel electrophoresis with radio-labeled bases. And here you can see that mutations were found in keratin 14, the primary keratin of the basal keratinocytes, and a hotspot at codon arginine 125 was discovered and continues to be the hotspot found today. Subsequently, the discovery showed that the partner of keratin 14 in these basal keratinocytes, keratin 5, could also be mutated and lead to this disorder. Further progress continued the next year, again by the Fuchs lab, in looking at another keratinopathy, the autosomal dominant blistering form of ichthyosis, called at that time epidermolytic hyperkeratosis, now called epidermolytic ichthyosis. And changes then were found in keratins that are found not in the basal keratinocytes, but rather in the suprabasal keratinocytes, where keratin 1 and keratin 10, the genes mutated in epidermolytic ichthyosis, are expressed. Subsequently, another blistering disorder. And I advise you to keep these ones in mind because I'll be repeatedly talking about the disorders I'm showing you now. This is called recessive dystrophic EB, an autosomal recessive disorder that's just a horrible disorder in which these children have generalized blistering that turns into scarring. It involves not just the skin, but the mucosal areas as well, and it eventuates in mitten deformities of hands and feet, as you can see here. And we knew from ultrastructural studies that, in fact, these children had an absence of a structural element called the anchoring fibril below the level of the lamina densa was where their blistering occurs. And it was Yoni Wito's lab that discovered thereafter these mutations in collagen 7 that lead to their deficiency. Followed then by work done both in Germany and here at the NIH, showing mutations in the gene that caused autosomal recessive congenital ichthyosis of the lamellar ichthyosis type. And this is a group of recessive disorders that tends to start, as this picture at your left, as a collodion baby, in which these babies are born surrounded by a collodion or saran-like thickened membrane. And over time, over the first month, that tends to clear and leaves behind a variably scaling disorder, as these examples show you. And this proved to be mutations in a gene called transglutaminase. And then the last disorder that I'll mention to you, one that was first described then in 2000 in terms of its underlying genetic basis, a disease called child syndrome for congenital hemidysplasia with ichthyosis and limb defects, with the defect being in a dehydrogenase of the cholesterol biosynthesis pathway. And this discovery in 2000 was the very first one among these to actually use Sanger sequencing to determine the gene abnormalities. And then you know the rest of the story. Over the next several years, Sanger sequencing really drove down the cost of doing these analyses, but still, the genome in its entirety was discovered using Sanger sequencing at a cost of $100 million. And then from there, starting around 2005, with the availability of new technology, second and third generation sequencing came to be and just escalated the output in terms of productivity in these discoveries and drove down tremendously the cost of doing this work. So we can be very excited in seeing the entire genome sequenced as shown here for a melanoma cell genome. But really, what we've needed and what has propelled forward the discovery has been whole exome sequencing. Now, the exome includes about 1% of the genome, but about 90% of the pathogenic mutations. And whole exome sequencing costs 10% of whole genome sequencing. So in a laboratory nowadays, one can do an entire whole exome sequencing for about $500. The beauty of this approach is that it's an unbiased approach to discovery. So one can simultaneously analyze all of the candidate genes, but also others. So you can find low-frequency mutations in known genes, mutations in newly associated genes, including in regulator genes, and you can also find low-frequency SNPs in thousands of samples. How has this helped our patients? And that is what I'm going to spend the rest of the time talking about. First of all, it has led to improved diagnosis. So just in the last few years with all these discoveries, we've seen new classifications for a wide variety of different types of genetic disorders of skin, including for the ones we're focusing on today, epidermolysis bullosa and the ichthyoses. Very importantly, it's led to less costly genotype analyses. Let's take as an example EB. A baby is born with EB, has blistering that's quite extensive, and we want to let the family know as soon as possible about the prognosis for this child, because the baby who has EB simplex, as shown here, tends to have a disorder that can be quite severe, particularly in the first few years, but subsequently tends to improve. And many of these children, by the time they're in late adolescence and beyond, barely have any issues with blistering. We can take junctional EB, a type in which most of the affected infants will die by the end of the first year of life. And then we can take recessive dystrophic EB, a disorder that we'll be mentioning again and again today, in which these patients tend to survive the neonatal period, but have a lifelong issue with severe itching, severe pain, and the scarring that I showed you before that is so functionally impairing for them. And in the past, just sequencing the collagen 7 gene that causes, in which mutations cause recessive dystrophic EB cost $12,000. Nowadays, we can concurrently sequence all of the possible genes through whole exome sequencing for just $3,000 for the patient. Another example is in the ichthyoses. I show you again, collodion baby, and then some of the faces of individuals with this type of autosomal recessive congenital ichthyosis. And Keith Choate at Yale has taken the developing new information about these various types and their underlying genotypes and has established a high-throughput multiplex amplicon for 11 of these most prominent genes. The cost for this now is $50 per patient for having this, at the same time, all of these looked at. And with this data, he's been able to look so far at 187 individuals with this phenotype and find all different genetic mutations among them. But what I point out that's most important here is that 20% did not have a mutation in any of these 11 genes. And that gives the opportunity then to perform whole exome sequencing and lead to gene discovery. Of course, for 2 decades now, we've been able to take DNA information and turn it into prenatal diagnosis to help our families. And this dates to the middle of the 1990s. I show you an example here of one of our patients with recessive dystrophic EB whose family did not want to have another baby unless they could be assured of having a baby who did not have this devastating disease. And we were able to use techniques like amniocentesis and chorionic villus sampling to perform a procedure in the first trimester that was very low risk and advise her that they would have a normal baby. And of course, in the near future, we don't have to have any risk at all because we'll be able to use maternal blood samples as a source of fetal DNA. Now, for those families who would not consider terminating a pregnancy and yet want to be assured of a normal baby, we can consider pre-implantation diagnosis, which has now been used for several genetic skin disorders, a technique in which in vitro fertilization will guarantee a normal baby through taking a single cell from the blastomere, extracting the DNA, analyzing it, and only implanting those fertilized eggs that will be assured of having a normal baby. Now, I'm going to spend the rest of the time talking about how understanding the underlying pathogenesis and obtaining genotype information can actually lead to new pharmacologic and new genetic interventions. And we're going to be covering a long list of examples that may be applicable to genetic skin disorders. And I'm going to start with the concept of read-through and specifically talk about that for one of the disorders I've introduced you to, recessive dystrophic EB. Aminoglycosides may read through premature termination codons to create a full-length protein. And it turns out that 10 to 25% of the mutations that cause recessive dystrophic EB are nonsense mutations leading to these premature termination codons. In fact, it's been shown that even though we know 50% is fine since carrier parents show no abnormality, even as little as 35% of normal collagen 7 levels should be enough to stabilize the skin. And in beautiful work done by Mei Chen's lab, cells transfected with constructs containing a variety of these null or nonsense mutations were tested, and gentamycin was able to induce up to 70% wild-type collagen 7 levels in these cells. Now, there is another way, another aminoglycoside for read-through that is actually available orally called PTC-124. You've probably heard of it. It's been used for other disorders. But unfortunately, that did not induce the collagen 7 expression. Now, topical gentamycin, though, is available, and we've been able to use that now in several of our patients with nonsense mutation with some good success. And I show you this young boy, and his cells actually were tested by Dr. Chen in these studies. And in fact, these studies showed that he could get 46% of wild-type collagen 7 levels when these cells were exposed to gentamycin. So he was certainly one of the first that we tested. And we found that when we topically applied gentamycin, it really expedited the wound healing in this boy's really otherwise recalcitrant wounds. One problem that we need to think about, though, is that when we're applying topical gentamycin to large body surface areas that don't have an epidermal barrier, there may be significant systemic absorption. And given the known renal and ear toxicity of gentamycin, we do need to keep that in mind, and we're limiting the application to the more difficult to heal areas. Now, another technique now is the use of recombinant proteins, and in fact, there's now available recombinant collagen 7 protein. In studies in recessive dystrophic EB mice, it's been shown that intradermal and intravenous delivery of recombinant collagen 7 increases the deposition at the wound basement membrane zone of collagen 7 and decreases fragility. And even topically applied collagen 7, when applied to wounds, incorporates into the basement membrane zone and can accelerate wound healing, not just in recessive dystrophic EB mice, but also in nude mice, suggesting that this could be a new treatment in general for wounds. I'm happy to say that the FDA approved moving forward for human trials with collagen 7 injection, and those will shortly be moving forward. I think one of the questions, though, that's raised by these is whether these immunocompetent individuals with recessive dystrophic EB who have been minimally exposed or not at all to collagen 7 will develop pathogenic anti-collagen 7 antibodies. And indeed, some recent work from Mei Chen and David Woodley has suggested that most patients with recessive dystrophic EB have anti-collagen 7 antibodies, but they're not pathogenic. We don't know what giving a bolus of collagen 7 protein will do, and something to be aware of. Now, sometimes replacement doesn't suffice, and I show you an example of this in these patients with child syndrome, as I mentioned earlier, the disorder with ichthyosis and the hemidysplasia and these limb defects, as you can see here. And in this girl who had this X-linked dominant disorder, orthopedists had to disarticulate that ineffectual limb, and so as you can see, she has an amputation at the knee area. Now, these patients tolerate anything topically very poorly, and we end up just using very bland emollients because there's never been anything to treat with. But this is an abnormality where there's a missing enzyme in the cholesterol biosynthesis pathway. So can we replace? Can we give cholesterol? And in fact, we can apply 10% cholesterol topically. Cholesterol is very important for skin, and it makes no difference whatsoever. But we can recognize through ultrastructural studies that, in fact, there is toxicity to the keratinocytes, and it appears to be the type that we see from accumulated metabolites. Fortunately, there is a rate-limiting enzyme at the very beginning of cholesterol biosynthesis, the HMA-CoA reductase, and we know that statins can inhibit that. So indeed, we put together a topical formulation of 2% lovastatin and 2% cholesterol to block the HMG-CoA reductase and to add the cholesterol end product. And I show you here in the girl that I showed you before and in yet another patient that there was dramatic clinical, histologic, and ultrastructural improvement over a period of just a few months. And I've now heard from at least a dozen patients from around the world in whom each of them has experienced improvement with this technique. And in fact, in the girl that I showed you, she's now able to wear her prosthesis and have a much more normal life because of this simple introduction of pathogenesis-based topical therapy. Now we can move over to cell therapy, and because the skin is so highly accessible, we can consider the grafting of keratinocyte sheets with gene correction to express the normal gene product. And we can think about this for our patients with EB. In fact, in 2006, a case was published from Italy in which retroviral gene-corrected grafts for junctional EB were applied. And in a follow-up paper more than 6.5 years later, they were still there and doing well. A study is about to start at Stanford in which collagen 7 corrected keratinocytes will be grafted for recessive dystrophic EB. Now, this is a first step. It's certainly not going to be the answer for these patients because there are many limitations. First of all, grafting can only be done to a limited area. Second of all, you just can't get the mucosae. And the mucosae in these patients that scar so much can be terribly incapacitating for them. A surgical manipulation in a patient who gets blisters spontaneously, let alone with trauma, can be quite risky. And then, of course, the issue, as with any type of these viral constructs with the normal gene, is the possibility of insertional oncogenesis. But one good thing you can think about is that if there is going to be a cancer, it's likely to involve the skin, be visible, and be amenable to surgical excision. However, this is a technique that is growing, and a recent paper stressed what is important, and that is choosing the clone you'll use wisely. Best to test one that has good stemness to maximally expand, and also exquisite safety, including assays of tumorigenesis in mouse models, and doing whole genome sequencing. Now, another technique that's actually been used for the last more than 5 years with some good results is stem cell therapy. It's been shown that when you take allogeneic stem cell transplants and you give them to patients with recessive dystrophic EB, and certainly before that with mouse models, collagen 7 will home to the wounded skin. In a study that was reported in New England Journal about 5 years ago, 5 out of 7 of these first patients were alive after more than 500 days, and many of them showed improved healing, shown here in a wounded area. At 180 days out, the deposition of collagen 7 at the basement membrane zone. And in fact, Jacob Toller and Wagner's group at University of Minnesota have continued these studies now with better results, reduced intensity conditioning, decreasing the risk, mesenchymal stem cells to increase graft take. And although still high risk, these patients are showing slow improvement, although the outcome is variable. An upcoming change to be considered is the recent discovery that one can repurpose AMD 3100, a CXCR4 inhibitor, which encourages stromal cell release from the marrow, and low-dose tacrolimus, which does that as well as seems to draw them to the skin, and in mouse models gives even better take and better wound healing results. And then we can't forget induced pluripotent stem cells, a very exciting option of using something highly accessible like skin fibroblasts or skin keratinocytes, making them into stem cells that can be then used to introduce after correction as a stem cell or as a graft. And the beauty of this technique is that one can use the patient's own cells and avoid immunosuppression. Of course, we have a long way to go before we can introduce these into patients, but there's some wonderful work being done. I also want to mention the possibility of using TALENs and CRISPRs as new approaches, because this is a technology, as you know, that can clear the mutant sequence and introduce the normal gene in such a way that it avoids random insertion and, of course, avoids the use of viruses. But one of the big problems with this is that it's still very low frequency of correction, and until that gets better, or until we can adequately clone out, we need to be careful about this technique. Now, I also want to move on to talk about dominant negative disorders, because here we have a different situation from what we've been talking about largely, where we can introduce something that is missing or is insufficient. Here we have a situation, as shown here, for the keratinopathies that we talked about, and as shown here for a dominant form of dystrophic EB, a milder form of what I showed you with recessive dystrophic EB, where the anchoring fibrils are fewer in number and are abnormal, so they don't function well, but certainly not as severe as not having anchoring fibrils. And in both of these situations, you have a normal allele with a normal gene product, and you have an abnormal allele that gives a full-length product, but it doesn't function normally, and it tends to lead to problems because of its incorporation. So we really want to target just that abnormal allele. And here's where techniques like RNA interference can come into play. And indeed, Sancy Leachman's group showed a few years ago proof that one could introduce an siRNA into human skin and cause improvement. In this case, it was an siRNA specifically directed against keratin 6A at a point mutation site in a disorder called pachyonychia congenita. Now, pachyonychia refers to the thickening of the nails, but the major problem in these patients is that they have exquisitely painful thickenings on the soles. And in fact, she serially injected greater amounts of this siRNA into a localized area on the sole of this patient, and as you can see, found improvement just at this site here, and on the other side, with the scrambled control, did not get that type of improvement. But the problem is that this injection was exquisitely painful, and the patient really couldn't tolerate going on with these studies, let alone somehow correcting the broader area of this genetic abnormality. So how can we painlessly deliver through the epidermal barrier that does such a good job of keeping out larger proteins and certainly oligonucleotides? And one exciting technique to mention is that of microneedles, a technique for painless delivery in which one can, particularly in a localized area, introduce a gene through that epidermal barrier. And this shows some work that's been done in which GFP was targeted, and as you can see, it disappears in the ones treated with the specific siRNA. Now, I want to introduce you as well to some of the work that we've been doing in conjunction with Chad Merkin, who's head of our Nanotechnology Institute at Northwestern. This involves spherical nucleic acid nanoconjugates, which are dense arrays of highly oriented oligonucleotides. They can be siRNA, they can be antisense DNA, which are spherically arrayed. And it is this spherical array that seems to impart the unique properties of these SNAs. There can be a central gold particle. We started out with some of those. And more recently, we've switched into liposomal cord or self-assembling structures of nanoconjugates. What's beautiful about these is that they can be delivered topically in common moisturizers. They're an all-in-one agent that requires no extra chemical or physical means in order to get through that epidermal barrier. And to date, there's been no evidence of any toxicity or immune or off-target effects. We've shown excellent gene knockdown in cell and mouse models and have published on the knockdown of epidermal growth factor receptor, and some work coming out this week in PNAS with ganglioside GM3 synthase targeted siRNA, and that depletes a lipid raft-based regulator of insulin signaling and normalizes diabetic wound healing in these models. Now, we can also achieve knockdown at the point mutation level, and I show you here an siRNA SNA directed against that hotspot mutation that I showed you previously in keratin 14 at the arginine 125 site. And you can see that with the scrambled, we don't knock down the green labeled abnormal mRNA, but we do knock down with the specific siRNA the abnormal, leaving behind the normal keratin structure of keratin 14. Now, I want to show you one more area that we're working on because I think it's particularly exciting and again shows the potential value of these SNAs, and this is targeting TNF for psoriasis. So tumor necrosis factor plays a key role in the pathogenesis of psoriasis, and we've now had for more than a decade anti-TNF monoclonal antibodies that we use for more severely affected patients with psoriasis, a disorder that affects about 3% of the US population. But these antibodies are costly. They cost about $20,000 a year per patient. And for the almost 90% of individuals with milder disease, there is no targeted topical therapy. So how could we consider using these SNAs? Well, we're now testing both liposomal and self-assembling SNAs. We've shown that all of these SNAs can pass beautifully through human skin, as shown here by this Cy5 labeled SNA. And we've gone on to show as well that it passes beautifully through the psoriasis human skin as well. We found that both the LSNA and the self-assembling TNF-SNA suppress TNF-alpha by more than 80% in these human psoriatic skin explants. And because there's about 90% homology of the oligonucleotide, the human SNA with the mouse TNF gene, we can start to do some studies in some mouse models of psoriasis. And I'll show you, just as a quick tease, some of the recent results we've had in an imiquimod-treated model of the development of a psoriasis-like eruption. And we've seen in this site-specific blockade of the psoriasis in this model by topically applied TNF-SNA. And I hope you can see here, we have on your right an area within the box that's been treated with both the imiquimod and the TNF-SNA. Surrounding it, you can see some erythema and scaling, and you can certainly see that throughout in the scrambled control treated exactly the same otherwise. And I think it's even easier to see when you look histologically. Here we have the TNF-SNA treated skin, and you can see that it really looks no different from the mouse who was not treated at all with the imiquimod, in contrast to the imiquimod and vehicle-treated mice, and in contrast to the mice treated with the imiquimod and the scrambled control. We can also look at transcriptomics, and here showing you some of the markers of psoriasis. We can see, again, the expected increase in TNF-alpha, in beta-defensin, and in the S100A7, and the suppression of loricrin, which represents the abnormal epidermal differentiation, and we can see no difference between that treated with the TNF-LSNA and our untreated by anything mouse. So I'm going to go on and spend the rest of the time talking about mosaic skin disorders. Now, gene mosaicism occurs when an organism is composed of two or more genetically different populations of cells that originate from a genetically homogeneous zygote. And postzygotic mutation is generally what causes this. Now, mosaicism may revert a disease phenotype to a normal phenotype by a spontaneous corrective mutation. This is called revertant mosaicism, and I encourage any physicians to be on the lookout for it because it can really help our patients. This gives them the possibility of natural gene therapy because we can expand then the patient's own cells from these revertant areas, whether for grafting or maybe in the future for making induced pluripotent stem cells, and use that. And indeed, I show you this patient here. This is one of my patients with recessive dystrophic EB. You can see her severe scarring. You can see the terrible deformity and contractures that she has on her foot. In fact, this was so bad that over the last few years she stopped being able to walk because of it. All she wants is to be able to walk again. But we've been tormented about the fact that we can't do serial casting because of the blistering tendency. How can we do a surgical procedure when she won't heal? But this girl and I found areas that never blistered on her. You can see this area on her back, you can see the area on her leg. And we were able to biopsy the area on her leg and show, in fact, that this area produced collagen 7. And working with Dr. Jacob Toller at the University of Minnesota, who has a GMP facility, we've been culturing her keratinocytes and her fibroblasts, and we're going to be able to use these phenotypically normal cells to provide a graft and move forward with the surgical procedure she so much wants. Now, mosaicism can also lead to localized skin disorders. And in fact, these disorders, as shown here by example epidermal nevi, were what led to Alfred Blaschko to describe lines named after him in 1901. And these lines of Blaschko seem to be lines by which mosaicism occurs when there is a change in the epidermal cell, and lines of embryologic ectodermal development. So you have a mother cell with a change and all of the cells that come out of that. are in that line and express that same mutation. But there really wasn't any proof of that. Now we know that if a somatic mosaic change just stays in the skin, it's just somatic, but it may also involve germline cells, especially if it's extensive. Now, when it affects germline cells, it can be a lethal problem, but it can also sometimes manifest as a generalized disorder. And we knew this in the early 1990s because of published cases of parents with epidermal nevi, as shown here, who had offspring with epidermolytic ichthyosis. And in fact, the biopsy from these epidermal nevi showed the same changes that we'd find in these individuals with generalized epidermolytic ichthyosis. So that led us to use that old technology with the gel electrophoresis to determine that indeed there were keratin 10 mutations at the hotspot of arginine in this gene, with a perfect correlation between the DNA sequence from the lesional versus the non-lesional keratinocytes, as shown clinically, as shown ultrastructurally, and as shown histologically. We did not see this correlation at all with fibroblasts, and this for the first time provided genetic proof that lesions along the lines of Blaschko represented epidermal somatic mosaicism. Well, now we have whole exome sequencing, and we can use this so much more easily to decode cutaneous mosaicism. And indeed, over the last few years, that's just what happened. We can now compare expressed DNA from a lesional biopsy using something like laser capture microdissection just to isolate the tissue that we want. And then we can compare it with genomic DNA from blood or saliva. There's been tremendous progress made in understanding these mosaic birthmarks or nevi that children are born with, whether we're talking vascular birthmarks, whether we're talking about melanocytic nevi, or whether we're talking about epidermal nevi. And it's been found that in each of these situations, these are localized sites of signaling pathway activation. Now here we have port wine stains, not associated with any syndrome, and then port wine stains in association with Sturge-Weber syndrome. And it was shown just in the last few years that about 90% of non-syndromic port wine stains and Sturge-Weber syndrome are caused by mosaic mutations of GNAC, in fact, at a hotspot in GNAC. And it turns out that these mutations in GNAC activate RAS-ERK signaling, as shown here. We can look at other vascular malformations like those involving the veins. Venous malformations show activating mutations not in that pathway, but instead in the PI3 kinase AKT pathway. These are activating mutations in angiopoietin receptor or TIE2, and again, they tend to occur most commonly at a particular hotspot. And most recently, we can look at lymphatic malformations and overgrowth syndromes like Klippel-Trenaunay syndrome, and find here that the mutation tends to occur in the catalytic alpha subunit of PI3 kinase, again, in that same pathway. And when we switch over to melanocytic nevi, we find that these all tend to be in the RAS pathway. So 80% of congenital melanocytic nevi, or moles, are postzygotic mutations in NRAS at a particular hotspot at the glutamine 61 site. And in other nevi, whether they're speckled lentiginous nevi or nevus spelus or Spitz nevi, these have activating hotspot HRAS mutations, particularly at the hotspot of glycine 13. And then we can switch over to these linear verrucous epidermal nevi, a different kind of nevi that don't show the epidermolytic changes that we see in the one I showed you earlier in that parent. This is a benign epidermal hyperplastic disorder in which one can see either activating mutations in one of the RAS genes or something along that FGFR3, PI3 kinase, AKT pathway, and one can't tell which pathway by looking clinically or histologically at these epidermal nevi. But once again, hotspot mutations abound, and you'll see that some of the hotspots listed here, like the RAS hotspots, are ones that we've seen before in the mosaic disorders I just talked about. And we can move on to some epidermal nevus variants, nevus sebaceous, nevus with woolly hair, nevus with bone abnormalities and hypophosphatemia, and see that these are all among the different RAS mutations, and again, with the same hotspots. In fact, this starts to explain disorders like phacomatous parakeratotica, a long word that basically means the combination, either in the same area, in some different areas in the same individual, of epidermal nevi and lentiginous nevi. And this is because there are HRAS mutations in keratinocytes giving way to the epidermal nevi and in melanocytes giving way to the lentiginous nevus. And then there's another one, phacomatosis pigmentovascularis, in which we see port wine stains in association with blue lesions like nevus of Ito, nevus of Ota, and Mongolian spots. And I bet that these all will be shown to share GNAC alterations because, in fact, mutations in GNAC have already been shown to cause most blue nevi, in addition to, as we discussed before, causing port wine stains. So finally, can we use this information therapeutically? Will topical, or for more extensive disease, systemic, RAS-ERK, or PA3 kinase AKT pathway inhibition improve these lesions? And although we still have more work to do, I would posit that we should be trying to go in this direction. In fact, rapamycin is now used topically to treat the facial angiofibromas of tuberous sclerosis, even though it's really a very large molecule. I'm not sure how it gets through skin, but it works quite well for these patients. And keeping that in mind and having a better understanding about these pathways, I tried it in a 4-year-old who had a lymphangioma that was very problematic for her on her neck. And we treated for 2 months with 1% topical rapamycin. And as you can see, this caused dramatic flattening of the lesion in this young girl. And there might be ways as well to block the RAS pathway because we now have available MEK inhibitors. And we get a glimmer of the possibility that this might work from a few studies, like this study which just came out, in which a MEK kinase inhibitor was administered 3 times per week for just 2 weeks to a transgenic mouse model that develops extensive congenital melanocytic nevi of the skin and of the brain and meninges, particularly in the first weeks of life. And as you can see in this model, there was a marked decrease in the pigmentation in skin, through the use of this MEK inhibitor. And here's another anecdotal example that was recently published, a patient who had clearance of both newly eruptive from vemurafenib treatment and pre-existing melanocytic nevi within months of the addition of a MEK inhibitor. So perhaps we can make a topical MEK inhibitor and use that for localized treatment. So I remind you that we have these small molecule inhibitors, and they may be useful, but we also have the possibility of gene suppression. And I will just mention that in our lab, we've now developed SNAs that can suppress FGFR3 and suppress HRAS, and we hope to move these forward as examples of other ways to treat these conditions. I also remind you that we've largely talked about congenital lesions or birthmarks, but we have so many acquired mosaic lesions as well. That mole on your arm, or as shown here, these seborrheic keratoses that so commonly develop in older individuals, those stuck-on brown lesions that one particularly sees on the trunk. And as it turns out from work that Christian Huffner has done, one can find these pretty much all in the FGFR3 signaling pathway with mutations pretty much all along the way. So I would posit that even these acquired mosaic lesions might be amenable to this new type of therapeutic intervention rather than a procedural technique. So in summary, this new technology has facilitated the advancement in knowledge about gene function and the effects of gene alteration in both genomic and mosaic skin disorders. These discoveries are guiding innovative treatment now, but even more so in the future, to reverse the phenotypic changes. And the decreasing cost and the increasing ease of decoding an individual's genetic change can lead to personalized pharmacologic or gene-based therapy. And I have a long list of individuals with whom I've collaborated and worked over the past few decades that I want to thank as shown here. And I want to thank you for your attention. Thank you very much.
Amy, thanks so much for a lovely talk. We have some time for questions. Please go to the microphones if you have a question.
Perhaps off topic, but I was wondering if you had any thoughts about pseudoxanthoma elasticum, PXE?
Thoughts?
A little bit perhaps out of left field.
Well, I think one of the issues with that is that we think that the skin lesions actually have to do more with a metabolic defect, at least from Yoni Wito's work. And as you know, there are studies going on right now looking at whether you can alter magnesium intake and try to work on that. Obviously, if you have situations where you need to replace something in the skin or you need to knock something down, we can use that as another example. There might be ways to move forward in that. But that's a little bit of a different disorder because we think what's going on in the skin may be more somewhat of a secondary phenomenon rather than a direct effect of the genetic change, but very good question.
Thanks for an excellent talk. Loricrin that you mentioned on one slide and several of the keratins are differentially expressed in oral cancer, and so I was wondering what's the applicability of your siRNA technique for that disease?
Well, what we have developed is the beauty of it is the potential for topical application through an epidermal barrier. So when we're treating oral cancer, we've got two problems. One is, well, it's quite as difficult to treat with technology because you don't have the barrier. But very importantly, number two, topical treatment, as you may well know, is very difficult because it's hard with oral, let alone with an ENT, head and neck type of cancer to do that. So that would be the type of thing that one would want to use a systemic approach. So there are systemic approaches being developed for RNA interference or antisense technologies that would do better in terms of staying around and hopefully with targeting sequences that allow one to home right to the squamous cell carcinoma in that particular area. But a topical approach, I think, would be very difficult for that.
Okay, how would this relate to adult lesions such as basal cell carcinoma or trichoepithelioma, et cetera, in terms of, we always think of these in terms of sun damage or some type of a damage, but how much could this actually be in terms of how you're, mosaicism in terms of how you're describing things?
Well, those are similarly mosaic disorders. With the basal cell, for example, the hedgehog signaling pathway abnormalities. And there are small molecule inhibitors, as you probably know right now, that block the hedgehog signaling pathway. So these small molecule inhibitors are coming out increasingly, and as we understand the pathways that are involved in these various types of benign or malignant tumors, we can start to inhibit it. You know that the problem with these small molecule inhibitors is that in some cases there may be a permanent change, but in other cases it may be temporary. And we could topically take an approach that would avoid all of the side effects, for example, that we've seen with the hedgehog inhibitors in patients, whether we're talking about the hair loss or the taste differences or the various other systemic problems that they've experienced by putting something on topically. But I think in some cases with malignancies, the question is going to be, do you want to just control it with that? If you use something that would block that pathway, would you want to couple it with something that would lead to an apoptotic situation or something that would totally clear the cancer rather than just suppressing? We know that when we stop the oral inhibitors, it's still there, it comes back. So that there would need to be work done to couple that with an agent that would not only suppress, but would actually kill it, so to speak. Now, one of the things that we've been working on in the lab in collaboration with Chad Merkin's group is also attaching chemotherapeutic agents that could be targeting cancers, so you can come in, suppress genes, but also at the same time selectively in those cancer cells kill them. And I think that sort of approach would have to be used for cancer.
Okay, thank you.
You covered quite a few different areas. In terms of the antisense therapy or siRNA therapy, there is no targeting. But since you have the lesion,
Right.
you could see, you could evaluate.
Right.
So for some of this, there is a saturation process. It needs some time for it to build up to prevent the formation of the messenger RNA that might be coding some of the genes for proliferation. So what is the delay period you find by topical application of some of the siRNA or antisense?
I'm sorry, I missed the word. What is the what?
What is the time delay? How long you have to wait?
Yeah. We've only used them in cells and in mouse models. And there is a little bit of a delay of a few days, which is presumably while we're suppressing the message and turning it into proteins before we start to see things acting with improvement over time that just gets better. I can't say we've treated any mice for more than about a month with these, but we do see sustained improvement in the ones that we're watching in the studies that we've done so far. We still don't know exactly what is the fate as we keep churning out these different SNAs, as an example, because that's what I had the most experience with. They certainly get stuck in endosomes, and some of them are in the cytoplasm. They don't get into the nucleus, but we've been able to show that they are serially cleared if we stop using them, so that after about 10 days after we stop putting them on, 98% of what we measure in the beginning are cleared. So we're still trying to learn about this, but it takes just a few days for them to start working, and then it does tend to turn around when they're lost if we just stop treating over time. And we really haven't done the studies yet to see how long some of this lasts. It's probably going to be very much SNA dependent.
Thank you. Good luck.
Thank you.
So Amy, you showed, I think, some very striking preclinical results with some of these SNAs and mentioned that you thought the structure was critical. Mm-hmm. Do you think that these are more stable or more potent or penetrate better or some of all of these things? Or do you
all the
have
above.
Additional comments?
Yeah, sure, thanks. So a lot of these studies have been done by Chad's group in terms of some of the in vitro work, but the spherical configuration has been shown to be much more stable, to have excellent cell uptake, and what we've shown is we've come in to look at the mouse skin and the human skin and to show the penetration. So when we're just using gold particles, they don't penetrate as well, but I think the major thing is that wonderful uptake and the greater stability. And modifications as well, for example, in the oligo structure can also help with that. So we've been gradually improving on the structure as well.
So are they charged? I mean, do they penetrate the stratum corneum well without additional manipulation?
Yeah, so they'll get right into the dermis when we look at them 48 to 72 hours later. But when we've looked in organs to see, because if it gets into the dermis, the possibility of getting into the bloodstream, we've seen a tiny bit, like about 5% of what we see in skin at most in the liver, and really nothing else appreciably in anything else. They're excreted through the gut.
Are there any other questions for Dr. Paller? Well, thank you so much. Please join. There's a small reception in the library immediately following the talk, so please join us for some coffee and maybe a cookie or two. Thank you.